Hey there, science enthusiasts. Today, we're diving into the microscopic world of bacteria and uncovering their secret weapon, hemolysis. Imagine bacteria throwing a tiny party on a blood agar plate. Their party trick? Breaking down blood cells. This process, my friends, is called hemolysis. Hemolysis is like a fingerprint for bacteria, helping microbiologists identify them. It's a crucial tool in diagnosing infections and figuring out the best way to fight those pesky microbes. So next time you hear hemolysis, remember it's not just a fancy word. It's a powerful tool that helps us understand and combat bacterial infections. Now let's meet the stars of the hemolysis show: alpha, beta, and gamma. These Greek letters represent the different ways bacteria break down blood cells on a blood agar plate. First up, we have alpha hemolysis, the shy one. Alpha hemolytic bacteria partially break down red blood cells, leaving behind a greenish discoloration around their colonies. Think of it like they're leaving a little bit of a mess after their party. The culprit behind many ear infections is a classic example of an alpha hemolyzing bacteria. Next, we have beta hemolysis, the showstopper. Beta hemolytic bacteria are the life of the party, completely breaking down red blood cells and creating a clear zone around their colonies. It's like they cleaned up their act perfectly. A well-known example is the one responsible for strep throat. And lastly, we have gamma hemolysis, the wallflower. Gamma hemolytic bacteria don't participate in the blood cell breakdown party. They grow on the blood agar without causing any hemolysis. Think of them as the polite guests who don't make a fuss. E. coli, a common resident of our gut, is an example of a gamma hemolytic bacteria. Now, where does all this hemolysis action take place? On a special plate called blood agar. It's like a delicious buffet for bacteria, made with, you guessed it, blood. Blood agar is a rich medium that provides everything bacteria need to grow and showcase their hemolytic talents. The blood cells in the agar act as the indicator, changing color or disappearing based on the type of hemolysis. Microbiologists use blood agar plates to quickly identify the hemolytic patterns of different bacteria. This helps them narrow down the suspects and make accurate diagnoses. So next time you see a blood agar plate, remember it's not just a petri dish with a fancy name. It's a powerful tool that helps us understand the secret lives of bacteria. Sometimes bacteria like to play hide and seek, making it tricky to identify them based on hemolysis alone. That's where our secret weapons come in: impregnation discs. These small discs are like tiny detectives laced with specific chemicals that can differentiate between closely related bacteria. For example, the bacitracin disc helps differentiate between beta hemolytic streptococci. The strep throat culprit is sensitive to bacitracin and shows a zone of inhibition around the disc. Other streptococci, not so much. Another example is the optokin disc, used to differentiate between alpha hemolytic streptococci. Our ear infection friend is sensitive to optokin and shows a zone of inhibition around the disc. Other alpha hemolytic streptococci, not a chance. These discs, along with other tests, help microbiologists unmask the true identities of bacteria and guide treatment decisions. It's like solving a microscopic mystery.